Welcome everybody to our product theater sponsored by Synovian. I'd like to thank Synovian and CPNP for putting this together. My name is Roger Somi. I've been active in CPNP for a long time, basically since it existed. I am a professor of pharmacy practice and psychiatry at the University of Missouri, Kansas City, and also the associate dean for our program in Columbia, Missouri. So I am joined today by two colleagues from Synovian, Dr. Mike Barber and Jennifer Wilbanks, and I'll have them introduce themselves, starting with Jennifer.
Hi, good afternoon. My name is Jennifer Wilbanks. I'm a pharmacist, and I've been with Synovian for about 6 years. I'm based out of Michigan, and I work in field HEOR covering the central region. And in addition to Synovian, I'm also an adjunct clinical professor at Ferris State University College of Pharmacy. Happy to be here. Mike?
Thank you, Jennifer. I'm Mike Barber. I'm medical science liaison with Synovian. I got my PharmD at the University of Oklahoma College of Pharmacy. Did my residency and fellowship in psych at the University of Missouri, Kansas City before embarking on a career at the University of Houston College of Pharmacy. Then moved into the industry, pharmaceutical industry, 21 years ago. Half that time I've spent at Synovian. So back to you, Roger.
Yeah. So Mike joined the industry when he was 12. That's why he looks so good. So today we're going to cover a lot of territory. So what we have to accomplish today is to look at drug discovery through the approval and post-approval process. Look at how these components help us translate that research data into clinical practice, and then we're going to highlight along the way some examples of innovations and interject some of our personal experiences with clinical trials. So you are all very familiar with this whole process where there is this stepwise approach to developing medicines, right? So we start out with, approximately 1,000 new discoveries, things that somebody thinks is going to be a putative medicine, and in the end, after they've gone through the discovery process and the preclinical work and the clinical work, we end up with one product, right? And so it's a lot of work and a lot of effort that's put into getting to that one product. So there's some estimates. If you look at the references here, this is really a study of like 100 different medicines that have come to the market and what the actual and opportunity costs were. And so those costs are estimated to be anywhere between 1.5 and 2.6, and probably really good estimates around $2 billion that it takes just to bring that medicine to be approved. But then you have to add on to that the costs that are associated with the R&D that is post-approval, and that can be several $100 million even post-approval. So and that's the average for all different kinds of drugs. And I believe that because psychiatry studies are a little bit more difficult, that the cost for those are actually even higher. So what we're going to do today is we're going to talk about discovery, preclinical, chemistry, clinical research, the FDA review, and then post-marketing processes that are involved in drug development. And so I'm going to make the transition here to Dr. Barber to tell us a little bit more about drug discovery.
Thank you, Roger. There are different pathways to drug discovery with the goal of getting an approved new molecular entity. You can identify a new biologic target, screen compounds without knowing their target that elicit a specific phenotype, or screen chemicals that target previously established therapeutic targets. We'll describe each of these today, as well as other ways to develop drugs, such as making new derivatives of approved drugs or developing new indications for existing drugs. In other words, repurposing them, as shown in these bottom right two boxes. Next slide, please. Let's take a look at each of these pathways, beginning with the identification of a biologic target or target-based discovery. So this approach relies on existing knowledge or discovery of a specific biologic target that's believed to be a key component of disease pathology, or it could be a mediator of response. There are myriad ways of identifying such targets, including postmortem examinations, genetic linkage to illness, etc. Once a target is settled upon, drug discovery seeks to screen compounds that bind to this target. There's an old saying in baseball that goes, see the ball, hit the ball. Once a compound is screened and is confirmed to hit the target of interest, and by the way, that target could be a receptor, an enzyme, or a mediator of signal transduction, then we go about finding out where that ball goes. The lead compound would then move forward along the development pathway with the hopes that one day it would become an approved new molecular entity. Next slide, please. So let's take a look at phenotypic screening. This might be a bit of a new term for many of you. So we define it as identifying lead compounds by selecting from chemicals that elicit effects and elicit their effects to ascertain whether or not they translate to beneficial impact on disease states of interest. So in essence, this is a target agnostic approach, and it means that you don't know the pharmacologic profile that mediates the effect, which is why you see, moving from left to right, target identification is actually in the middle of this pathway after it already has become a lead compound. I'll talk a little bit more about that in just a moment. So next slide, please. A very specific example of phenotypic screening is depicted here. So the assays that are used to assess a phenotype for a chemical under study typically employ bioinformatics, in which vast amounts of biological data points undergo computational methods to provide useful statistical information. So this would be too massive of an undertaking for humans. So we train computers to use machine learning techniques by which algorithms are written and trained. They examine relationships of millions of observed data points and present the relationships in ways that we can understand and visualize. So this helps us to screen millions of data points for trends and relationships to targeted processes for the signature of a drug as a potential target. So you can see that moving from left to right, you get to that signature and you kind of make inferences about what that chemical might do. So this process that's specifically depicted in this slide involved proprietary hardware owned by a collaborator using an in vivo mouse behavioral platform that measures spontaneous and evoked behaviors such as locomotion, body posture, and behavioral sequences. So behavioral signatures for hundreds of marketed or clinically validated reference drugs were characterized to produce a library of drug class signatures for assessing novel molecules. So the goal in this case was to identify compounds with antipsychotic activity that do not exert their antipsychotic effects via direct binding of D2 or 5-HT2A. That's called an anti-target approach. So you don't know what the drug does, but you know what it doesn't do. So no D2, no 5-HT2A. As it turned out, one of the compounds that had been screened using this procedure, it had a signature depicting similar activity to known antipsychotic compounds. And this is how CEP 363-856 was identified as a lead compound. Next slide, please. In terms of targeted approach, let's take a historical real-world example of a targeted drug success story using a genotypic approach. The discovery of the Philadelphia chromosome and BCR-ABL, supported by the National Cancer Institute, demonstrated for the first time that a genetic alteration could cause cancer. In the 90s, Dr. Brian Drucker of Oregon Health and Science University hypothesized that leukemia could be treated with a drug that blocks BCR-ABL specifically. So unlike chemotherapy, this targeted approach could kill the cancer cells but leave normal cells intact. So in working with Nicholas Linden, who led a drug discovery group at a pharmaceutical firm, the pair of scientists identified a compound that was remarkably good at targeting chronic myelogenous leukemia cells, and it was better than any drug they'd ever investigated up to that point. So that drug, STI-571, later renamed imatinib, is a small molecule tyrosine kinase inhibitor which blocks the abnormal BCR-ABL protein that signals cancer cells to multiply. So imatinib was the first therapy targeted towards leukemia. This breakthrough led to a new frontier in cancer treatment, often referred to as personalized or precision medicine. So now it gives me pleasure to hand the virtual microphone over to Jennifer, who will provide more examples of drug development.
Next slide. And thank you, Mike, for that. So yeah, I'm going to go on to how some of the other compounds are go through the different pathways. So this slide, it talks to the therapeutic targets specifically. So we know that antagonistic and agonistic properties of certain receptors causes certain type of therapeutic responses. So these two medications on the left-hand side, the tendaspirone and the perospirone, these are medications that, psychiatric medications that were approved in Asia. And knowing that the agonist activity at 5-HT1A is anti-anxiety, that's where tendaspirone was discovered. And then adding the D2 and 5-HT2A antagonistic properties gave it the atypical antipsychotic therapeutic mechanism. So knowing both of that and wanting to develop a product that had some of those similar properties between these two molecules, that's where lurasidone was discovered and obviously is currently marketed, exhibiting some of the same properties, but also different properties that weren't exhibited in the original molecule. Next slide. So when we look at new active ingredients, it's defined in the US as a new chemical from an active ingredient that's already marketed today in the United States. And I'm not going to spend a ton of time on these examples because I know, we're talking to a group of pharmacists today. So I'm sure that you remember some of these in your pharmacy school days. So isomerization is basically where you're taking the chemical structure and you're modifying it. So you're getting different results, you're doing different things to the structure and the configuration, and that may provide different activity on the molecule level. So the example here is in, asthma or COPD, but if we look at the psychiatric space, another example would be the drug citalopram to escitalopram. And I know there's probably a ton of examples that you could look at for each of these different types of development pathways. Looking at prodrug development, so a prodrug in itself is inactive and it's not converted to the active molecule until it enters the body. And there's a number of different reasons why you may want to do that. For example, by using a prodrug, you can improve the delivery of the active molecule to wherever the site that you need it to work. You might be able to decrease some of the adverse events. Or as in this example here, the dextroamphetamine and lisdexamfetamine, you might be able to decrease the abuse potential by delaying its effect by a certain amount of time. And then lastly, we look at active metabolites. So this example here, also in the psychiatry space, we're looking at risperidone. When a drug is metabolized, you end up with metabolites, and those can either be active or inactive. The paliperidone is the major active metabolite of risperidone, with some differences from the parent compound. So, sometimes the metabolites may have better efficacy, or they may have different adverse events. Their pharmacokinetic and dynamic properties can differ from the parent compound. And so that's where this really comes into play in the drug development pathway. Next slide, please. So drug repositioning is an example of where you have a current marketed product, you have the original indication, and there, during the course of use of that particular medication, you might find that it's effective in other areas. And so these are just a handful of examples of that. I like to use the first example, the minoxidil, because I remember that from my pharmacy school days, where it was originally indicated for high blood pressure. But in use of that drug, they found one of the adverse events was hair growth. So then it was studied for and approved for hair regrowth. Some of the other examples, like botulinum toxin, is a product, a molecule that has been repurposed numerous times. And again, I'm sure there's a number of examples that could fall under this category. Next slide. So if we look at new formulations and new routes of administration, I'm going to provide just some examples in the treatment specific to Parkinson's disease. So levodopa has been around for a long time. You're familiar with its use with carbidopa to be used for maintenance therapy for Parkinson's disease. But if you change its route of administration, it's also being used today as a continuous sub-Q infusion and also as an inhalation powder for different indications, still within the Parkinson's disease space, but for it's indicated for different time points within disease. Amantadine is another example. It can also be an example of a repositioning because amantadine was originally an antiviral, which was found to have some Parkinsonian effects. And so just changing the formulation of that extended release versus immediate release, and again, using it maybe in different time points in that disease state. And the last example is apomorphine. Again, apomorphine has been around for a long time, originally approved as a sub-Q injection, but now approved as a sublingual film. And if you go to the next slide, you can see here that it was recently approved again. It's a non-ergoline dopamine agonist, and it's indicated for the acute intermittent treatment of OFF episodes in patients with Parkinson's disease. So same molecule as the sub-Q injection, but this is a sublingual film that dissolves under the tongue. Next slide, please. And then lastly, just talking about drug repurposing, I went over a few examples of this. Another example may be where you have a combination of products that maybe it's one pill instead of maybe 2 or 3 separate pills. That's also an example of repurposing. And that can be important from an adherence standpoint. If you think about patients and having to take multiple products throughout the day versus having to take one product, it can be helpful from that standpoint and having patients remember to take their medications. And then if we look at the different review processes, the FDA does allow for almost a different type of pathway for these repurposed agents. It's a 505(b)(2)A NDA. And what that really means is that some of the information that might be coming from the sponsor for the approval was not actually conducted by the sponsor. So it could have been information that was previously conducted maybe by the original manufacturer who had that original indication or the original approval, but some of that information is used and presented to the FDA for the approval of the different product or the different indication. And now I'm going to turn it over to Roger to go over the preclinical research. Roger?
Thanks, Jennifer. Yeah, so we've had a couple of products come through in psychiatry in this 505B approval process. And what's interesting to me is that I think one of the reasons it was developed was to make things go a little quicker. And in fact, if you look at the vast experience with the 505B strategy is most of those approvals take a little bit longer. So there are some advantages in terms of numbers of patients that get exposed to a potential product that doesn't work, but it's not really a cost saver or maybe a cost saver, but it isn't a time saver. So just going to briefly discuss some issues around preclinical research as the molecule begins to come into development. So there's extensive preclinical study that lead to some of the early evidence in terms of efficacy and toxicity and learning about the pharmacokinetics, the drug metabolism, and some of the early safety data. It really gives us some idea of what kind of doses that we might want to give to patients. Some of this is done in vivo, some of it is done in vitro. There's a lot of cell cultures, test tube kinds of research that's done. One of the things that I always thought I wanted to do as part of my background was always have a test tube full of D2 receptors sitting on my desk so I could really understand what I was doing when I was doing the work with patients with schizophrenia. But there's lots of studies. There's actually a recent review of how the mice, of mice, just mouse models of psychotropic drugs, right? And the numbers of different things, the in vivo receptor assays, the behavioral assays, you know, we look at cataplexy in dogs, cataplexy in mice to predict EPS, for instance, and those really sort of are the models that Mike was talking about earlier that really sort of form this, what do we think this drug is going to be when it grows up kind of concept, and is this going to be a safe molecule to give to humans, right? So it really sort of sets the stage for a lot of the clinical work. And once, and not necessarily simultaneously, but in concert with is this work that's done from a chemistry and manufacturing control point of view that really sets the stage on developing a product that can be used in sufficient quantities and sufficient and given in a way that will work for our clinical trial work. So it's really critical as you're looking at registration trials to assure the quality and stability of the investigational drugs. So there's a lot of controls that go into this phase of the drug development. There's the early work thinking about, you know, what is this drug going to look like from a formulation point of view? Is it going to be a capsule? Is it going to be an injection? Is it a tablet? What's the best way to present what's currently a powder to the patient? There's a lot of controls, right? Controls for temperature and moisture, light exposure, all kind of, you know, depending on what that product is. I remember one clinical trial that I did. There's a lot of controls that are developed around this, the packaging, et cetera. One of the trials that I did, this was a product that was an injection, and it would ship to me. I would get my drug shipment for, as I enrolled subjects into the study, in a box that was about half the size of a refrigerator. And in that box, the styrofoam was about 3 inches thick and then there was another box inside that box, and inside that box was more freezer packs. And inside that box was 12 small vials of drug, right? So imagine a box shipping to me. Inside that box were temperature control that I had to turn off and record. And if there were excursions from the requirements, I had to record those and impound the drug. I mean, there's a lot of stuff that goes into making sure that the drug is actually getting there. And I was thinking, man, if we got to go through this every time, that's going to be a big problem for pharmacy in terms of getting this drug. But when the drug actually hit the market, it came there with no refrigeration requirements. So there's a lot of ongoing things that happen in this area as well. So this is something that just isn't done completely prior to all the clinical work being done, but it's an ongoing issue. So the next step really is to work on the clinical studies, right? This is where the IND comes into play, the investigational new drug application. And so there is a lot of studies that are developed by the company that's working on this drug. And those studies are developed in consultation with the FDA, in consultation with FDA guidance, and they're very specific with each protocol, and lots of different studies are developed, right, that look at lots of different things, right? So you need to think about your patients, duration, what are your control groups going to look like? Are you going to have an active control? Is it just going to be placebo controlled? What are the assessments? Are those assessments things that the FDA will accept to show efficacy, and is there anything unusual that came out in the preclinical work that you'll need to do in terms of safety and toxicity studies. So the IND is submitted, and it's an agreement really between the FDA and the company about how you're going to develop this drug. And so it sets out all of that previous data and the studies that you're proposing for the to develop the drug, and the FDA reviews that, and if it's approved, then we move ahead with the various phases of human research. Right. So the first phase, or phase 1, right? So this is a early, right? So the scale of the studies, it's early. Really, what we're trying to do is to find what's the safety, how much can we give, what kind of pharmacokinetics exists. So we'll learn something about the half-life here. We'll learn something about metabolites. It's generally done in less than 100 people, and it's done fairly quickly. These are the studies that you get advertised. We'll pay you $3,000 to stay in our place for a week, and what actually happens is you go through a study that's depicted here, right? So this is an example of 2 different formulations of drug. It could be a food interaction study. It could be all different kinds of things that you want to know prior to, developing, or you're bringing this drug into the second phase, which is more patients with the illness that you're targeting. These are usually healthy volunteers that move through this. And so they might get a single dose on day 1, a single dose on day 2, right? It's in this particular study, it's a crossover, immediate release versus extended release formulations. And so it helps you decide, do you want an immediate release? Do you need an extended release? And about a third of the drugs don't make it through this phase. Right, there's toxicity that happens, right? So we are beginning to, weed out drugs here, right? So a lot of drugs, come out of phase 1 and then they enter phase 2. So phase 2, there's oftentimes the phase 2A and 2B. So these are really sort of pilot studies or proof of concept, right? The 2A studies really are, does it look like it works? And the 2Bs are what dose seems to be the right dose for this population of patients. It begins to give give the new molecule to several hundred patients now. And this can last for several months, up through, actually, we've had patients who entered phase 2 studies, and then there was a long-term extension study that we could enroll them in, and they were on it until the drug was developed, years later. So, they can last for long periods of time. Usually the data the first analysis, the first cut is weeks to months worth of exposure to the drugs. And we lose about a third of the drugs, or we lose about 2/3 of the drugs that came into phase 2. And so as we move into phase 3, right, if we look at the statistics on just numbers here, we started out with 1,000 molecules that somebody thought might have been a drug, and we enter, if you're the drug company and you tested 1,000 molecules, you are now down to 3, right? And so what moves out of phase 3 is about 1/3, right? So this is where one drug comes out, right? And so this is more of a large scale, where we've used the data that we learned in phase 2, and we are exposing larger groups of patients that actually have the illness that we're targeting. These can be, there's usually a fixed dose portion of it. It can be, it's oftentimes active and placebo controlled. Some of the challenges with that particular design is that the data is oftentimes difficult to analyze. Those of you who've heard me speak on this before, you know, all we're trying to answer is does this dose work? Not is this dose better than another dose? So for instance, in this particular study that's shown here, we're not trying to answer the question, is 120 milligrams better than 40 milligrams? We're just asking the question, does 40 milligrams work? Does 120 milligrams work? And you put the active control in there to try and make sure that your experiment is working, right? Because these are all experiments in the end. So almost all of these studies have this open label extension. The purpose of the open label extension is to get data on what happens if you give patients this drug for extended periods of time in terms of safety. So these are mostly safety studies and not necessarily efficacy. Efficacy would be a secondary endpoint for all of those studies. So once all the data is in, we have preclinical data, we got the clinical data submitted to the FDA, and I'm going to turn it over to our FDA expert, or at least of the three of us.
Maybe among the three of us. Thank you, Roger. So, you know, clearly so much work has been done to this point, as has been described by Roger and Jennifer in today's presentation. So now it's time to show your work. And by that, I mean submission of all of this data for review. So as we move into this slide, we'll take a high-level look at the arduous process that the FDA must undertake in order to fulfill its mission of ensuring that safe and effective drugs are made available to improve the health of people in the United States. So if anybody's listening from the FDA, I hope that sounded nice. So once the phase 3 trials are completed and the sponsor feels that they have enough evidence to go before the FDA, they must submit the NDA or new drug application. So this includes, but may not be limited to, proposed labeling, chemistry, manufacturing, control information, as Roger described earlier, clinical data and clinical data that also Roger described earlier, human pharmacology and bioavailability, safety data, patient information, all the stats, statistics. If we have any experts on here, it would blow your mind to see the amount of statistics that you do to look at entire packets. And the FDA is very sophisticated with their own statisticians. So and case report forms generated from all the supportive pivotal studies. So all of this gets put together in a single package and sent to the FDA. So the FDA has up to 2 months to decide if the packet is complete, at which point they will respond to the sponsor and either accept the submission, meaning that they will review the drug, or they will issue a refusal to file letter. That'll detail the reasons why they do not consider the packet sufficient for full review. But once it is approved and review is underway, the standard review process typically takes 10 months. And there are, of course, 2 outcomes for that review. The FDA will notify the sponsor of an approval, or they will issue a complete response letter, or CRL. The CRL informs the sponsor of the reasons that resulted in their conclusion that in the current form, the drug application does not warrant approval. So I mentioned 10 months. Let's take a look at some of where this comes from in the next slide. Prior to 1992, timeliness of the FDA drug review process was a big concern. So the Prescription Drug User Fee Act of 1992, we refer to it as PDUFA, it added user fees to provide funding for the FDA staffing and other resources that ultimately benefit the payer of the fees, i.e. the sponsor. So as you can see in this slide, we're now at the 6th version of PDUFA. So when the sponsor hits the send key to transmit the NDA to the FDA, that transaction basically costs about $3 million. So this was increased about 14% in 2019. And this is if it includes clinical data. There are applications that only cost $1.5 million if you're not including clinical data. That may be something like a labeling change due to AEs, and all of this funding that's coming from that initial submission accounts for 20% of the FDA's revenue budget. So 80% of their funding comes from prescription drug program fees. So that's an annual assessment. If you have an approved drug in the FDA Orange Book, you have to pay $325,424 per drug on October 1st of each year, and that's up to 5 drugs. So if you have 5 drugs in your portfolio, you pay $1.5 million per year to keep those drugs listed in the FDA's Orange Book. Next slide, please. While PDUFA resulted in a more streamlined, predictable review process with a time requirement to get to that final decision efficiently, there are several designations within the FDA that can also shorten the review and or approval process. So you see those listed here. I'm not going to go into great detail, but beginning with Fast Track, the FDA may approve this designation if the therapy fills an unmet need and a variety of conditions, including showing superior effectiveness and things of that nature. And examples of recent Fast Track designations include remdesivir for the treatment of COVID-19 requiring hospitalization, and also one maybe more that hits close to home with CPNP folks is cannabidiol for the treatment of seizures associated with Dravet syndrome in patients 1 year of age and older. Breakthrough therapy designation may be granted by no later than the end of phase 2 if the drug is, again, for a serious condition and has preliminary clinical evidence indicating substantial improvement on clinically significant endpoint over available therapies. There are other possible requirements that might lead to such a decision, but of course it does help streamline the process for those who are designated in such fashion. A recent example of a drug that got that designation is the one I talked about earlier, SEP 363856, which is intended to treat patients with schizophrenia. Accelerated approval, the FDA decision on that is based on positive results from surrogate endpoints such as laboratory measurement, radiographic images, physical signs, and things of that nature. They're thought to predict clinical benefit, or they serve as an indicator of a very early indicator rather than going to an irreversible morbidity or mortality outcome. So, for example, tumor shrinkage versus overall survival. Confirmatory studies are required even with accelerated approval by the sponsor that those take place in phase 4. A recent example of a drug that has gone through the accelerated approval pathway is the recent approval of aducanumab to treat patients with Alzheimer's disease. And in terms of priority review, the FDA's goal of action is within 6 months of a submission if it increases effectiveness in treatments or diagnosis or a variety of other factors. And again, that gets you to a shorter cycle. Instead of the 10 months, you get 6 months review. A relevant example of a drug, this is not so recent, but let's look back to 2007 that got priority review, was the supplemental new drug application for aripiprazole in the treatment of adults with major depressive disorder as adjunct to antidepressant therapy. So that was a very high level view, but keep in mind, a lot of these sound familiar. A drug can qualify for more than one of these categories, and people get them mixed up all the time. So I hope that this will at least somewhat clear up some of the differences between these distinctions. So Roger, could you move to the next slide, please? So the involvement of the FDA, of course, does not end with drug approval. So let's take a look at basically what happens next, which is phase 4. Regarding safety surveillance, the MedWatch program is a gateway for reporting problems with approved drugs and learning about new safety information. Submissions can be made by healthcare practitioners, doctors, etc. The sponsor, which I may add is required by law. Anytime an employee of a sponsor learns of an adverse drug reaction, whether through an HCP, a news report, or a family member, we're required to submit those. Also, consumers, of course, may report adverse events through MedWatch. So a newer program that got underway in recent years called the Sentinel Initiative. It started out as a mini pilot, but it has since expanded. Sentinel uses very large electronic health databases, for example, electronic medical records or EMR systems. They use administrative and insurance claims databases and registries, and it's intended to monitor the safety of approved medicines in real time. They use machine-based learning and algorithms to pick up important safety signals. And an example of one of the first things that came out of this the initiative by the FDA Sentinel program is the boxed warning for depression associated with the asthma and allergy drug montelukast. So Roger, let's move to the next slide, please. So the FDA Adverse Event Reporting System, or FAERS, is a database that contains the adverse events reports, medication error reports, product quality complaints, and anything that resulted in adverse events that are submitted to the FDA. So that database is designed to support the FDA's post-marketing surveillance program for drugs and biologic therapeutics. FAERS is searchable. Caveat, the FAERS website has a message that you may not be able to interpret your search findings, and the FDA says that they're happy to help you understand your search. Our advice is don't expect something to pop out that is user-friendly unless you're a statistician and have worked with these types of reports. Roger, do you have anything to add here?
Yeah, I think this, the example of montelukast and the depression warnings and suicidality warning is really important because it shows the role that non-researchers, clinicians can play in how drugs are used and how they're labeled and the regulatory process. So it's not just the researchers that are part of this process. It's everybody. I mean, we're all in this together. So.
Absolutely. Great points, Roger. Thank you. So there are many other things that can happen in the phase 4 post-marketing phases. Supplemental applications, dosages, formulation changes. The INDs, for example, a schizophrenia agent may be looking for a new indication for bipolar depression, could move from adults to kids and things of that nature. Next slide, please. Occasionally, the FDA will have questions that it wants answered, even while the approval takes place. So the examples could be including drug effect in children, establishment of the lowest effective dose, things of that nature. So the approval letter will provide details of these phase 4 commitments, as they're referred to, that the sponsor must fulfill as a condition of the approval. So these are generally large, simple trials that are unblinded, observational, retrospective, post-hoc analyses of data that you already have, or case control series. So Roger, what's your experience with this kind of work?
So we've done a lot of phase 4 work. We've done studies where we had an interesting question, and we went to the company and said, "Hey, would you fund a study looking at this particular issue?" And that's the investigator-initiated studies, the IIS or IIT process, and most of those run through the MSL. So you guys get to see a lot of those and work with us on those. I've also done some post hoc analyses where, if you've got a question that you want to have answered, you can oftentimes work with the medical section of the company and get them to do a reanalysis of some of their data. I presented, several posters on this over the years at CPNP, and that leads to publication. So this is a way for just the regular Joe kind of guy to be involved in the work, and we've also done a lot of the clinical trials that were required by the FDA. Once the drug hit the market, and so I think the next slide is an example of one of those requirements that the FDA have.
Sure. Thank you, Roger. Let's talk about an example. And earlier I talked about that they can be pretty simple, these post-marketing commitments, but in this case, it didn't seem to apply as much. The FDA was not convinced that 20 milligrams was not proven to be ineffective in adults with schizophrenia, since the only study that used 20 milligrams in the submission had an assay sensitivity arm of haloperidol, and that did not separate from placebo. So essentially that study was not really convincing of anything. So what you see depicted here in the top and bottom arms was determined whether 20 milligrams was superior to placebo. That said, the sponsor went above and beyond in order to answer an important clinical question, which is, what do you do at 2 weeks if the patient hasn't improved? This involved re-randomization based on whether or not there was an early response in order to determine whether there would be a benefit to increasing the dose versus leaving the dose as is in those early non-responders.
So I just want to chime in here, Mike. Sure. And for me, this is really important data, because the fixed studies that come out of phase 2 and phase 3 are hard to apply to clinical practice, and they don't have Yeah, so well, what about? You got a lot of what about questions, and so these phase 4 studies really help us, answer those questions and get to that, how do we dose this drug in our patients now that we have access to the product? How do we dose the drug? Because people are going to do all kinds of weird things, as we know, once the drug hits the market, and the more evidence that we have, the better decisions we can make in the clinical arena.
Thank you. That's spot on, Roger. I appreciate those comments. And so all of that work that we've talked about so far really indicate whether drugs are safe and effective, which is wonderful. But there's a lot of choices out there about how to treat patients. And sometimes people wonder what is the value of a particular drug. So I'd like to turn it over at this time to Jennifer. And Jennifer, you could please walk us through the study of health economics and outcomes research.
Sure thing, Mike. Thanks for that. So up until this point, we've talked about the different pathways to FDA approval. And I just want to spend the last few minutes here talking about HEOR. The slide deck really was meant to go over the drug development, and the next few slides is by no means meant to be a deep dive into HEOR, but just to give you a brief introduction to it. So HEOR really has come about because, as Mike said, there are a lot of alternatives that are available to treat various disease states. Obviously, the healthcare costs have risen. So we really want to look at these products and the new services that come available and compare them to what's currently available and whether or not they're cost effective. So HEOR really is looking at the costs and the consequences of both products and services. And it always involves a comparison between the two. Really, it answers the question, are we receiving the greatest healthcare benefit for our dollar? HEOR is meant to complement the traditional evidence-based information. It's meant to be used as a guide for those decision makers who have impact on patient access. It's not meant to be the decision maker, but meant to be a guide. And there are a number of different outcomes that we can look at with HEOR information. We can look at the economic information, obviously, the monetary outcome. What may be the cost avoidance for using a particular product? Can we decrease hospitalizations? Can we decrease length of stays? We can also look at the clinical benefits. So is a particular product that may be new to the market, that may be more expensive, does it decrease the number of deaths in that particular disease state, or is it curative of a particular disease state? Or it can be something as simple as, looking at markers. So for diabetics, does it decrease the HbA1c? Or in cardiovascular disease, does it have some impact on blood pressure? And then lastly, looking at the humanistic perspective, obviously the patient's experience with care is very important. What is their health-related quality of life? Do we have an improvement on their functional status? Can we help these patients become more productive in the workplace for a longer period of time based on how we're treating their disease state? Next slide, please. So there are different time points to have those discussions with the FDA as far as patient-reported or humanistic outcomes. And so this slide is just giving a summary of when some of those discussions can occur. And you can see that depending on what type of outcomes that we're looking at, those discussions can happen quite early. If you look at the green shaded, as early as the start of phase 2. So what the FDA recommends is that if you're using some type of outcome that may be new, or if you're modifying a patient-reported outcome, then you wanted to start those discussions with the FDA early in the process so that there can be ongoing discussions and agreement as far as what the final outcomes will look like. If you're using something that is already in place, maybe we don't have to have those discussions quite as early with the FDA. But I think the FDA really is recognizing the importance of the patient's perspectives and their outcomes and everything, because you're seeing more and more patient-reported outcomes being reported out in the clinical trials and being used as an endpoint, really to try to differentiate these different therapies. Next slide, please. So as I mentioned before, the information that comes from HEOR can support post-marketing decisions, really meant to be used again as a support tool, not as the decision maker, but it really does help answer the questions on all these different fronts as far as, how is a product going to be covered? Will there be any utilization management tools applied to it? Will it affect your healthcare policy? Will it affect some downstream costs or maybe the way you manage certain disease states? And again, it really is answering the question, is the extra benefit, the extra outcome that the potential positive outcome that you may be getting, is it worth the extra cost? And I put an example there on the bottom right-hand slide where there was a study that looked at the treatment of ventilator-associated pneumonia, where vancomycin was a standard therapy, standard of care, relatively inexpensive. And then a new product comes to the market, linezolid, which was obviously more expensive from a product standpoint, but how did that differ in the outcomes? And what the study found was that linezolid, even though it had a higher upfront cost, you had better outcomes for the patient on the back end. So improvement in their the rate of improvement and decrease in their hospitalization stays. And at the end of the day, it was more cost effective to use this more expensive medication up front, just because you could decrease some of those costs in the long run. Next slide. So just wrapping this up, when we talk about real world evidence, what is it? And this is a direct quote from the FDA, that it's information on healthcare that is derived from multiple sources outside of the typical clinical research setting, and it includes the real world data. So we know that information that's collected in the course of a normal healthcare, whether it be electronic records or claims, it can be very different from what happens in the controlled setting of a clinical trial. So it's very important to collect that information just to see what happens in the real world setting. And from there, real world evidence is that clinical evidence about the usage and potential risks or benefits of a product that's derived or used from the real world data that's collected. So this is most probably frequently used in the decisions for formulary placement, whether or not you're going to have some type of whether it's a prior auth or step therapy. And then the FDA cited real world evidence trial, again, signaling that the FDA is recognizing the importance of having this real world data. In this real world trial assessing the dose response, it was an adaptable trial, which was a patient-centric study that looked at the benefits and long-term effects of 2 different doses of aspirin on secondary prevention. So it was a large study, long-term, that really looked at how outcomes differed in the real world setting. And so with that, I'll turn it back over to Roger to bring us home.
Thanks, Jennifer. That was a good review of really how the work continues after even all the FDA requirements are being met, to look at where this drug fits. And I think all of that is really, really helpful, but it generates just a ton of data for us to look at. So to summarize, there's a huge investment of time and resources, and there's lots of different roads that we travel down to see where for these new products to be developed. And clearly, there's lots of regulations, as Mike pointed out, that need to be addressed to ensure the safety and efficacy of the product before it comes to the market, as well as after it goes, after it hits the market. And there's opportunities for us as pharmacists to be involved in the ongoing aftermarket monitoring, some of the health economic outcomes that Jennifer talked about that can impact the labeling. We've done studies that have changed the label that are phase 4 studies that, is in a specific population or a specific clinical challenge that ends up changing the labeling of the drug. So it's all really, really good in terms of helping us understand how to use these drugs in clinical practice and what the impacts are from an economic and health outcomes point of view.